Okay, I misspoke a little bit. I got one more topic before mutations. There's one more little interesting thing about eukaryotic transcription and translation that is noteworthy, and it's different from prokaryotic. When it comes to eukaryotic transcription and translation, there's actually a step in between the two that involves modification of the messenger RNA. And what happens is we have sequences in the RNA that are actually taken out, and removal of those is what actually creates the final, what they were sometimes referred to as the mature RNA molecule. So you essentially have sequences in the first, immediately after transcription, that is, you essentially have sequences that have to be removed. And so you have another group of enzymes called spliceosomes that actually cut out the parts that need to be removed and then join the parts that need to be put together. Why this is done is that they're pretty sure that what this essentially does is it allows unique gene combinations. So you can actually splice these up differently, and you can actually take one piece of RNA prior to splicing and end up creating multiple unique combinations after splicing. So they think this is a way that the cell even gets even more variation from its proteins. You can get different unique protein combinations by splicing up the genes differently. So anyway, that's kind of why they think this happens. But what's confusing about this, and what's absolutely backwards, is that what they do is they call the parts that are removed, they call them introns. And they call the parts that are kept, they call those exons, which makes no sense. You can think awkward naming. I don't even know what to say about that. I lost my train of thought. The point is that you have parts that are cut out and parts that are kept in, and they essentially named them backwards, and I have no idea why. So the introns are cut out and the exons are kept, and that's somehow that made sense to somebody. I tried to look it up one time, couldn't get an answer for it that made sense. So anyway, here you can see parts of green cut out by the spliceosome, parts in red combined back together to make the full mRNA, the mature RNA, excuse me. So it's basically an extra step that the cell goes through. So if we go back real quick and just kind of visualize this here, you once got one more time DNA to RNA and RNA to protein. Okay, transcription, translation. So in between here and here, you get the spliceosomes that come along and process that DNA to make a unique or mature form of the messenger RNA. One of those weird things that happens in biology, and again, why they think this happens is it creates more variations of the same type of gene. So you can essentially take one gene and from that information rearrange it to get 10 different protein combinations from it. And that's partly how they think organisms can get more and more complexity from a smaller amount of DNA. And without going into too much detail, I will tell you that one of the most surprising things about studying the human genome, which was only concluded, or really began, I should say, kind of concluded and began at the same time in around 2003. 2003 was the time they fully sequenced the human genome and just finally started looking at it in a way that they could make sense of it. And one of the things that they found was that there was only about 20,000 protein coding genes in the human genome. And that really baffled people. No one expected that. The estimates initially were in the millions to hundreds of thousands, and people were literally dumbfounded. Scientists were literally dumbfounded that the human genome contained only about... Now, initially they estimated it much higher. It was initially estimated at closer to 30,000, and since then that number has come all the way down to about 20,000. So it's not an exact science. But the idea is that now we recognize there's only about 20,000 genes, which is very few compared to what they thought it would be. And so the ability to create unique combinations from the same gene is thought to account for that increase of variation. So you really end up getting more proteins than what it looks like just from the number of structural genes. So that's kind of how they think this plays a significant role. And I don't know if that makes sense, but that's kind of the idea behind eukaryotic transcription being different than prokaryotic transcription. Okay, now we'll take a look at mutations, 9.4. Mutations are any kind of change in the DNA sequence. And the idea is that when we have a change in the sequence, we can have a change in the information. And that information constitutes, or change in that information constitutes a mutation. Now, when you start messing with the genetic code, you have a real ability to change protein structure and ultimately cause them to be non-functional. So a lot of times mutations will affect the structure of a protein, and that change in the structure of the protein will affect some function in the cell. Now, sometimes the mutation is relatively harmless, the effect is very minimal, and sometimes the effect is catastrophic and it can be fatal. These here are kind of colloquial examples I pulled. They're just kind of like when people think about a mutation, this is kind of what they think about. The truth is, this is not really the common types of mutations. But these actually are mutations. This one's actually thought to be induced by a chemical that people use on their dog called Frontline, that they think is a mutagen that builds up in wetlands and causes four-legged frogs, which is a real thing, by the way. What exactly causes it is hard to say. But four-legged frogs, that's a real thing. Two-headed snakes, that happens. There's two-headed cows, two-headed people. They're rare mutations, but they are. It's a change in the sequence. We actually have genes in our body that tell us where the head goes and how many heads that we're supposed to have. Isn't that crazy? Your genetic code literally says you have one head and it goes here, and then your eyes go here, and your nose goes here, and your ears go here. And if anything happens to that information, you can get an extra head. Suddenly, the DNA basically says, okay, now you have two heads, so make two heads. It's amazing how specific the DNA is. It tells us everything. And if you mess with that code, all sorts of stuff happens. Now, honestly, most of the time, what happens from a mutation is either cell death or cell cancer. Those are the two most common outcomes. So cancer is actually a type of mutation. But other weird things can happen. Here's another weird example. This happens in fruit flies called Ctenopodia. It's a scenario where a mutation actually causes the legs of the fruit fly to grow out of its eyes. So you have another gene that in the fruit fly that says your legs go here, and when you mess with that gene, the legs can end up coming out of the eyes. So anyway, these are all just kind of examples of some of the more crazy mutations out there. Now, a mutation, again, not typically that obvious, but a mutation is really kind of, again, it's just defined as a change in the sequence. When we talk about the effect of a mutation, we can refer to an organism that's affected as a mutant strain. Again, that's if they live, that's if they survive. Again, a lot of times a mutation takes place in a way that the organism dies. For example, if a mutation takes place during development, such as during the embryonic stage of life, then most often, especially if it's a significant mutation, the embryo is terminated and the pregnancy is aborted. So a lot of times a mutant strain never really comes to fruition. But that's not always the case, and there are a number of examples where a mutant strain does survive. In fact, there's lots and lots of examples, so it certainly doesn't always work that way. So when you have a living mutant strain, you call it a mutant strain, and really you can compare that to everything else that's not a mutant as a reference to the wild type. So wild type basically refers to the most common form, and a mutant is a deviation of that. So mutant strains will typically show variation in a variety of things. If they live, and if it's a mutation that affects the way something behaves or looks, we can have, for example, changes in morphology, nutritional characteristics, genetic control mechanisms, resistance to chemicals, even differences in environmental preference and enzyme functions. As an example of resistance to chemicals, you can think of antibiotic resistance. So how they look, how they feed, how they activate genes, how they become resistant to certain drugs, what kind of temperatures they prefer, and how their enzymes function are all subject to change, and more. But those are some of the ways that mutants can alter themselves if the mutation doesn't cause death or something like that. So mutation is something that, again, is a change in the genetic sequence, but it can be caused by a variety of factors. And there's two general classes of what causes a mutation. One's called a spontaneous mutation. The other is called an induced mutation. A spontaneous mutation is something that takes place due to errors in replication. One of the things that happens about DNA replication is that it takes place very, very fast. Now, I didn't get into that. I didn't talk very much about DNA replication, but it takes place really fast. And the speed that it takes place at means that it is subject to certain errors. So over the time of evolution, organisms have kind of found a middle ground where they can replicate their DNA fast, but not too fast to get too many mutations. So there's kind of an acceptable number of mutations that an organism can have, and that kind of coincides with the fastest speed that they can have for replication. So basically, all organisms will make small mutations during replication. Some will make more than others. So, for example, bacteria replicate faster than eukaryotes. So prokaryotes actually replicate almost 10 times faster than eukaryotes. And as a result, they make more mutations, but they're less susceptible to them because there's so many bacteria that if one cell dies, there's another cell to replace it. So anyway, spontaneous mutation is really just a change arising from replication or errors in replication. Prokaryotes make more mutations in this way than eukaryotes do. Induced mutations are a mutation that results from environmental exposure, chemical agents, and things like that. So this is a situation where something in the environment reacts with the DNA that causes it to be disrupted, and then the sequence is changed, and as a result, the effect is that the outcome, the characteristics or whatever of an organism change along with it. So a mutagen is kind of any one of these chemicals that can cause a mutation, and some of them are very weak and some of them are very strong. So there's a spectrum there of different intensities. I'm not going to get into too much on the specifics here, but as an example of chemical mutagens, you have basically molecules that can disrupt the DNA. One example is called an acridin dye. An acridin dye is a chemical that inserts between nucleotides and literally distorts the DNA double helix. Distortions in the helix will cause errors in replication, kind of like replicating too fast. So it's just another way to cause errors in replication. Another way is called a nucleotide analog. This is actually a molecule that looks like a DNA molecule, but isn't quite the same. So here's an example. I pulled this off the web. These are all examples of nucleotide analogs, and you can see they look similar to the base of like an adenosine. They have a similar structure as a normal nucleotide, but they don't fit the same way into the structure of the DNA molecule. So while they have a very similar nucleotide structure, what that really does, it just allows them to be uptaken into the DNA molecule well enough to be inserted, but not well enough to function correctly. So once inserted, they go on to cause disruptions in replication and even disruptions in transcription, and that causes mutations and other errors in errors in transcription. So analogs are mimics that are good enough to get inserted, but not good enough to function the same way. So they cause mutations. So those are some kind of general classes of chemical mutagens. What actually are the most common mutagens in general are actually called physical mutagens, and most of these come from the sun. They're basically these little particles that come from sunlight, and they have enough energy to physically strike our DNA and cause damage. Now, some of these are X-rays. They can come from an X-ray machine. X-rays are something that give off these high-energy particles. Gamma rays can actually come from nuclear radiation. This is why nuclear waste is really harmful. It actually gives off a strong degree of physical mutagens. And UV light is one of the most common sources of a physical mutagen, and this is one that comes from sunlight. All three of these are actually the same kind of particle called a photon, and each one travels at a different velocity. X-rays at the highest, gamma rays the next highest, UV light just below those. But each of them carry a huge amount of intensity in terms of a momentum of a particle speed. And when they hit something, they literally go right into the cell and cause damage to the DNA. Here's a quick video, not the best, so I was trying to find a better one, but here's a quick video showing...
DNA is under constant attack from...
This is showing DNA, the graphics aren't that good, but it's showing a bundle of DNA inside of a cell, and then it's showing some of the ways mutagens can take effect on it.
Reactive chemicals and natural
only about
background radiation. Free radicals are the byproducts of normal metabolism in human cells. Seen here as bright particles, they sometimes react with DNA and cause chemical changes. Radiation can also affect DNA. For example, ultraviolet light from the sun can cause harmful chemical changes in the DNA of skin. These changes can lead to kinks in the DNA that prevent genes from being correctly read, or deletions that alter the type of protein produced. Thanks to constant biochemical repair work, most mutations are corrected before they have any effect. But in rare cases, mutations can accumulate, and this can give rise to diseases such as cancer.
Okay, so not the best way, but not the best animation, but the idea is that DNA is literally being bombarded by particles coming from outer space, from the sun. At least that's the most common source. That's UV light. And what literally happens is that UV light is traveling at such a high velocity that these particles literally go through your skin and can hit your DNA molecules within your skin cells. And when they hit the DNA at just the right spot, they can actually cause a kink, and that can actually then result in a mutation. So, by the way, this is what, this is what's referred to, this is what can create, what's called a pyrimidine dimer, or what's known as a thymine dimer. So remember that thymine is a type of pyrimidine. And what happens here, and this is just one of those weird fluky things that just so happens to occur, is that where UV light hits a certain area, you tend to get a certain type of mutation that's called a thymine dimer. So when UV light hits in a certain location where two thymines are located next to each other, it tends to cause those two thymines to cause a kink in the location and in the space between them. So it causes them to form a bond actually between themselves, and that causes the entire DNA molecule to bend and bulge right there. So essentially what UV light does is it causes the DNA to bulge up and bend and twist, and that ultimately can lead to errors in replication. Now, amazingly, our cells actually fix these repairs and actually replace them in the correct form. It's something I don't get into as much anymore, but the video kind of alluded to that, and there's a number of videos out there if that's something you're interested in. You can look that up. But the body actually repairs itself when it does this. Now, as a side note here, I don't usually get up on a soapbox too much, but I will say, when it comes to sun tanning, it is now generally recognized that this is borderline stupidity, because you're basically giving yourself mutations at a really high rate. And the same is true of smoking. So, really not intelligent behavior, because what you're basically doing is you're flooding your body with mutagens. And these mutagens will physically cause damage to your DNA, which can lead to things like cancer. And really, I don't really get into this too much, but cancer is really a type of mutation. And it's a type of mutation that leads to a cell dividing abnormally. And it's one of the few types of mutations that you actually become aware of if it doesn't kill you, early on during development, I should say. most of the time mutations don't create color changes. Most of the time they don't make new shapes and patterns and characteristics. Most of the time they kill the cell. One of the rare things that happens where they don't kill a cell is that they cause the replication of the cell to become abnormal, and the cell actually stops dividing normally, and it actually grows and grows and makes copies and copies and copies when it shouldn't do that. So abnormal replication and cell division is one of the few things that happens with a mutation that doesn't lead to death. So there's lots of things that mutation can do. Most of the time it either kills the cell. Cancer is one of those situations where cells divide abnormally. So anyway, indoor sun tanning by a lot of the experts is really being looked at just like smoking is. And we all know today smoking is bad, so if you do it, so be it. Life is short. But the idea is that you know it's bad and you know there's a link to cancer, and if you say, screw it, I'm going to do it anyway, then more power to you. But at least you know that link. I think most people are kind of starting to figure out sun tanning is kind of in that same ballpark, but just to clarify, it's in that same ballpark. And that the Skin Cancer Foundation has pretty clear statistics on it. If you want to read those, I put those here. I'm not going to test you on that, but it's something that you definitely want to be aware of. Now, with both smoking and sun tanning and any exposure to carcinogens and cancers and mutagens, it's worse with age. So as you get older, smoking is more likely to cause cancer, so is indoor sun tanning. When you're young, you can pretty much get away with it without too much worry. So really, smoking and tanning, the effects become exponentially worse than at an older age. And really, you see a lot of skin cancers and things like that. Lung cancers typically develop later in life. So the body slows down and it's not able to repair itself the same, and that's when those effects get worse and worse and worse. So okay, so this next part here gets into what happens when you have a mutation. So a mutation is a change. There's different ways that it can change, different causes. So what happens when it changes? So first of all, we'll start with what's the severity of a mutation. So for example, we have what's called a point mutation, missense mutation, silent, nonsense, and frame shift. Each one of these has a different context in terms of what happens. Now, let me go back here real quick. I skipped over this first part. I apologize. The first part here is indicating that the intensity or the size of a mutation, whether you change one base pair or 1,000, can actually end up with the same result. So it's possible to have a small mutation that is just as harmful, sometimes even more harmful, than a larger mutation. So any tiny change can have a major impact on the DNA. Sometimes a small mutation is less harmful, but the truth is small and large don't necessarily correlate to more or less damage. So potentially any change can cause damage. So one of the most common types is called a point mutation. A point mutation is simply a change in any one nucleotide base pair. So anywhere you have an A and a T or a G and a C. Something happens there, that's called a point mutation. A point mutation then can essentially be an additive, deletion, or substitution, where you either add an extra one, so you basically fit an extra nucleotide into the sequence, you remove an extra nucleotide, or you remove a nucleotide, or you switch the wrong one. You replace an A with a T or a G with a C or something like that. So additive, deletion, substitutions. Those are the three things that can happen at a single point. A missense mutation is a mutation that ultimately leads to a change in the amino acid sequence, or changes any of the amino acids in reference to their codons. So if we have a mutation that causes a change in amino acid sequence in even at least one amino acid, then we call that a missense mutation. So a point mutation can oftentimes lead to a missense mutation. Now, sometimes we have what's called a silent mutation. This is where you have alteration, but not a change in amino acid sequence. And how this is possible is, let's go back one more time to this table we showed. And this actually goes back to another aspect of redundancy. Notice again, looking at serine here, UCU, UCC, UCA, UCG, all code for serine. And if you note, the first two base pairs of all 4 are identical. They all start with UC. The only difference is in the fourth one, UCAG. So the idea is that if we have a mutation in one of these spots here and it changes it to any of these other sequences, we still get the same amino acid. And so if we have a mutation, for example, that changes UCU to UCC, we have a mutation. It's a change in the sequence, but we still get the same amino acid. So it's referred to as a silent mutation because of the fact that we don't change the protein sequence. That is a silent mutation. So point mutations, single point of change. If that leads to a change in the amino acid, we call it a missense. If it doesn't, we call it a silent mutation. The next part here is looking at some of the ways that we can affect it from a larger standpoint. One of the most abruptive changes you can have is two of the most abruptive changes you can have are called nonsense mutations and frame shift mutations. A nonsense mutation is where you change a normal codon into a stop codon. So if we look at this figure, we can see that UAC, for tyrosine is very similar to UAA, which is a stop codon. So if we change that one little letter C to an A, what that does is it stops protein production wherever that mutation occurs. And this is an example where one tiny little mutation can have a devastating effect on that particular gene. So one change here, nothing really changes. One change here, and we basically stop the whole thing short. So here's a figure I pulled off the web. Here's the initial sequence. Here's a mutation at this spot, and we end up creating a stop codon here. So wherever this protein, wherever the mutation occurs in the sequence, protein production stops there. So you would get an incomplete protein. That would be very likely to cause an issue in the cell. Now, it depends on how important the protein is. Some proteins are really critical. Some proteins aren't that critical. So I like to think of proteins as like body organs. Some proteins are like your fingers. You can live without them. Other proteins are like your heart and your brain. You can't live without them. So if a nonsense mutation takes place with a critical protein, that's going to cause death. Sometimes it may not be as big a deal if it's a lesser important protein. But that's a nonsense mutation. Frame shift is where you have a change in the reading frames. Let me pull up another figure for this. So here's a figure showing a frame shift mutation. So here's an example of a normal sequence, ACG, AGG, ACU, etc. And these code for the amino acids here, threonine, arginine, threonine, alanine, tyrosine. We add one little letter. So this is an example of where an insertion mutation can cause a major change or a frame shift. What happens is when we add that one little letter here, yeah, they added one, right? Is that what they did? It's not very clear what they did here. It looks like, I'm sorry. They added a sequence. It's not obvious what they did now I look at it. But basically, we add a nucleotide, and what this ends up doing is it shifts the entire reading frame. And now the triplets are not in the correct sequence. So now we're essentially grouping them wrong. And now as a result, the different groupings translate to different amino acids. So the sequence doesn't look the same anymore. So now you get the amino acids arginine, glycine, leucine, histine, threonine, not the original sequence. So that basically constitutes a massive mutation. You've basically changed the entire sequence of the protein. Anywhere this frame shift takes place, you will shift all of the reading frames downstream, and it's likely that, like a stop codon change, you'll have a massive change in the protein. So these are generally some of the most lethal and massive types of mutations. But to clarify, they can take place with a single additive or deletion type mutation. So if you add or delete a single nucleotide, it can throw off the entire reading frame and cause each amino acid to be different than what it was originally. Here's a better figure. I forgot this was on here. I apologize. I'll let you look at this. This is actually from table 9.5 in the text. Take a look at this here. I'll pause it here if you need to, and it goes through different types of mutations here. Okay, here are the review questions for part 4. We'll do one more part and we'll wrap this up. This is going to turn out to be a much longer lecture here. So go ahead and pause, take a look at the parts for questions for part 4, and then we'll take a look at the end. Okay, so the next part, we just want to talk real quickly about the effects of mutation. Generally, as I mentioned, mutation is bad. It can lead to issues. A lot of times it can cause death of an organism, but there's a number of mutations that are more subtle. And over the extremely long history of humans and other animals, we've amounted a number of actual genetic mutations that are passed down from one generation to the next. To really understand this, you have to kind of get into Mendelian genetics and how dominant and recessive genes work. And we're not doing that in this class. Again, that's more of a principles of biology topic. So it may not make a lot of sense as to how that works from a genetic standpoint, but there are recessive genes, and you can have different combinations of recessive genes. And most organisms have only one or no recessive genes. But occasionally when two organisms mate and they each have a recessive gene, occasionally they'll pass those both on to an offspring who will get two recessive genes and have a disorder associated with that. So that's how you can get diseases that are passed down from one generation to the next. Like Huntington's disease is one of the notorious examples of a genetic disease that you can have in your DNA that you don't even know about until later in life, and then it's fatal. So some of that stuff gets kind of weird. Now, from a more traditional standpoint of how mutations affect an organism in the more short term, most of the time they are harmful or neutral. Harmful meaning that they're bad, as I mentioned, cancer, death, things like that. A lot of times they can be neutral. Not every protein is critical. Sometimes you can have an effective protein and have virtually no effect on the cell. There's not a lot of redundancy built into cells. They have multiple ways of doing the same thing. And a lot of that is to mitigate the effects of mutation. So in a lot of cases, a mutation won't necessarily cause any real effect on the cell, any noticeable effect. So this is why some exposure to UV light isn't going to kill you, and this is why you can smoke and not die of cancer immediately. A lot of times the mutations that happen really don't do anything. But it is random. Mutations are random. You're affecting DNA randomly. And so the more you affect it, the more you expose yourself to mutations or mutagens, the more likely it is that eventually those will cause a harmful effect. So you're kind of playing with fire, so to speak. But most of the time they turn out to be pretty neutral. Now, on super rare occasions, a mutation can actually turn out to be a good thing. Now, these are a little harder to explain, but especially as organisms reproduce and create more and more offspring, mutations in the offspring can sometimes turn out to be a positive thing. And I'll give you an example of that here in a second. Another thing about mutation is that whether it's good, bad, or neutral is often relevant to the environment and other organisms around them. So here's an example with using a bacteria as an example. If you have a mutation in an enzyme that is responsible for the digestion of lactose, this can have more or less of an effect depending on where the organism is. So to clarify, I'm talking about an enzyme that helps a microorganism digest milk. And this is actually part of what happens with lactose intolerance. You get bacteria in your gut that start breaking down lactose instead of your body doing it. But anyway, bacteria can break down lactose. They need an enzyme to do that. Now, so if a bacteria gets a mutation that causes that enzyme to stop working, they can no longer metabolize lactose. Now, if that organism is in a highly nutrient abundant area where they've got 1,000 other nutrients besides lactose. You can have glucose and sucrose and fructose and maltose and dextrose and blah, blah, blah. There's 1,000 other nutrients besides lactose. You don't necessarily need lactose. It's just something that you can use. So if you have a ton of other nutrients, that mutation may not really have a huge effect because it just basically shrugs it off and uses a different energy source. However, if that organism is in an environment where lactose is the primary nutrient, suddenly that organism can be in big trouble, especially if it's going to come down to having lactose or not getting energy. So if that's the case, then that's a mutation that's going to be fatal to the cell. So really, harmful and neutral is a lot of times a consequence of what's being affected and then how the environment then affects that the outcome. So this is just a kind of an example I made up to show how in this particular case, lactose being one of many nutrients may not always be a problem unless that's one of the few, and then you got a real problem. Now, when it comes to mutation, every population of a species will be measured and observed in science to have some level of mutation. So within any population, you have, say, for example, less than 1% that have some type of mutation. Now, really, any mutation occurs at about 1%. So really, almost every organism has some kind of mutation, to be honest. It's just a matter of whether it's obvious or a problem. But within any population, you have mutants that occur usually at a small frequency. Now, most of the time, mutants are not necessarily a good thing. A lot of times they die off, or it's just not necessarily a big deal, or sometimes it's kind of only a minor deterrence for them. Here's an example that's used in a lot of biology discussions, and it's kind of a classic example. I even titled it that here. It's one of the few examples where you can see the effect of mutation in the short term. This is a real field example that they pulled from the early 1900s, around that area. What they had was an area where you had different colors of moths. There were a group of moths called peppered moths. Most of them initially were a white kind of peppered moth that looked like this. But you occasionally had black mutants that looked like this. Okay, so normal wild type black mutant here. What happened in this particular example was they were studying these moths in an area that was experiencing what was called industrial melanism. Industrial melanism is a process by which pollution from the environment causes tree trunks to actually darken. And it was a real issue back in the early 1900s because they didn't have hardly any environmental regulation. And these factories were just pouring out toxic metals and all kinds of crap. And they were literally just darkening trees. And that was just, the tip of the iceberg. But in this particular example, darkening of the trees was where the effect came in. So these peppered moths, as it turns out, they tend to sit on these trees, and the wild type under normal conditions, if you look here based on this figure, the wild type actually would normally blend into this tree fairly well. The pattern that it's evolved to have actually matches and camouflages it with the tree trunk, which is what you'd expect. The mutant doesn't blend in as well. And the problem for that mutant is that it's actually easier prey. So there's birds that feed off of these, and birds are visual predators. So they're actually looking for these color patterns. And this one stands out, and the idea behind this is that as a black moth on a light colored tree trunk, you're easy prey. And so the initial population of these moths was much higher. It was a much higher percentage of peppered moths and a very low percent of black moths. So the wild type common was peppered, and this was a very low frequency black moth. Okay. Now, what ended up happening with industrial melanism, as I said, is it changed the tree trunks darker. Okay, so what ended up happening was it literally changed the environment around these moths, and now you had a darker tree trunk that was actually better for the darker colored moth and now worse for the light colored moth. And this was kind of a weird example because of human influence. The tree trunks change within a matter of years. Normally the environment doesn't change that quick. It takes thousands if not millions of years. But this was an example where you saw rapid change, and you actually had people studying these moth populations. So it was actually kind of a good example of where, short-term change and direct observation coincided, which is not something that a lot of times happens, so especially short-term change. But this was an example that allowed scientists to measure it. So what they were doing is they were measuring populations while this industrial melanism was occurring. And what they found was that as the trees got darker, the populations literally shifted, and that the white or the lighter colored peppered moths, they actually went down, and the black colored populations went up, and it shifted dramatically in huge swings. And then later, as the pollution went down and the environment was restored, it shifted back to the wild type. So what it really showed was that being light colored or a darker black was only good in relation to the environment. If you're in a light colored forest, this is the ideal color. If you're in a dark colored forest suddenly, that's no longer good. This is actually good. So this is an example where being a mutant turned out to be a benefit, and it actually over time made this group of moths the more common type, at least for a short period of time. So that's just kind of a weird example of how mutations can actually be good. Now, when you look at the odds and the probabilities, I like to think of a beneficial mutation as like a winning lottery ticket. The odds are not good, and most organisms will never experience such a thing. So they tend to occur over long periods of time, given many rolls of the dice. So kind of like a lottery ticket. Billions will play, and only one will win. So you can think of a beneficial mutation as kind of similar types of odds. Most of the time, a mutation is not good. Antibiotic resistance, I mentioned this one. Antibiotic resistance is an example where you have rapid mutation because of rapidly growing populations. Now, bacteria are known to mutate very rapidly. They replicate so quickly that there are guaranteed to be mutations within every population, and these populations then can kind of randomly generate mutants that can sometimes generate beneficial traits. So in a population on your body, for example, you'll have, give or take, several tens of trillions of microbes. Let's say 50 trillion bacteria in your gut. 50 trillion, okay? If only one of those has a mutation for an antibiotic, and you take that antibiotic, then theoretically that one mutant can survive the antibiotic and then regrow to create a whole new strain of bacteria that are also resistant. I'm going to try and wrap this up here. So if there's lots of videos on this, I'd encourage you to go find one that's got an animation here. But the idea is that there's so many that when you start introducing an antibiotic, it's basically a guarantee that one or a couple of them are going to be resistant to it just naturally through natural mutation. And so you're basically just selecting any bacteria within that huge population that has a trait that favors that drug. It's almost like saying we want to find some person somewhere in the world that has some weird, totally odd talent. And you can probably think of almost any weird, crazy talent, and somebody out there in the world can do it. And there's only 7 trillion of us. I say only 7.5 trillion. But with that many people, somebody will fit almost any crazy, weird description or talent as the example. So with 50 trillion bacteria, the same odds kind of apply. You're eventually going to have one or a couple that are resistant to almost anything or can kind of fit almost any weird exception. So when we start using antibiotics, we're basically just saying how long is it going to take before we find the mutants in this population that can survive, and really the question is, will they be able to repopulate with a whole new mutated strain? So once you eliminate all the affected bacteria, those who are not resistant, what you end up doing is allowing room for the antibiotic resistant forms to grow, and then they start to grow in the absence of other competing bacteria. So that's where antibiotic resistance and mutation go hand in hand. Lots of bacteria and lots of chances for mutation events that cause resistance. Last part here, we're going to look at a couple of unique examples that take place only in bacteria, and they do what are called recombination events. And this is where they essentially swap DNA with each other, and not through replication. Most of the time DNA is transmitted, it's through what's called vertical transmission from parent to offspring. What bacteria are capable of doing is what's called horizontal transmission. So let me clarify here. Sexual reproduction is the primary means of distributing your genetic material. So as eukaryotes, we sexually reproduce, and we actually mix up our DNA between maternal and paternal DNA. So a male and a female mix their DNA to create an offspring that is a combination of the two. So it's a way for, more advanced species to mix DNA. Bacteria and simple-celled organisms, they don't reproduce through sexual reproduction. They actually reproduce asexually, which simply means they copy their DNA and make identical versions of their initial cells. So really, they don't have nearly as much variation. They're typically just a replica of the previous version. What these organisms have evolved to do, though, is they found ways to increase variation by swapping genes between different populations and shuffling chromosomes back and forth to create new gene combinations. So it's a way to get around the monotony of just copying the cell over and over and over again. And so the idea is that they're called recombination events. It's a situation where one bacteria literally swaps DNA with another bacteria. Anytime that happens, it's called a recombination. One of the most common ways this happens is with an extra chromosomal DNA called a plasmid. We talked about plasmids way back in chapter 4. It's a little piece of DNA that is transferable. And so what ends up happening is these segments get swapped back and forth. This type of example, I'll come back to that here, by the way. This type of example is what's called horizontal gene transfer. The transfer of DNA from one adult cell to the next. Let me just pull this up here. So let me just put a quick diagram together here. So here's our initial parent cell, and vertical transmission would be DNA being passed down the traditional route through replication. So the cell replicates, it creates two daughter cells, and the DNA from the parent is passed to these daughter cells. So that would be, to clarify, vertical transmission. Horizontal transmission is when the DNA is transferred from one parent cell to essentially another parent cell. It's not really the right term there, but it's basically from an adult cell to another adult cell. So it's basically being passed to another cell in the environment that isn't a direct progeny of that cell. So essentially what's going to happen is it's got these little pieces of DNA, and it's going to replicate that, going to make an extra copy of it, or at least one extra copy, and then that extra copy can literally get transferred over into this other cell over here. So any sort of trait or any characteristic or any phenotype associated with this DNA will be passed from one cell to the other. So if this is again DNA, and the DNA contains traits, any trait on that DNA gets passed from the initial cell to the next. This is not something that we see in nature very often, just to clarify. This is fairly unique and not something that you see in higher level organisms, as far as people can tell anyways, as far as scientists can tell. The equivalent is something like swapping the ability to play basketball between two people, or like swapping hair color back and forth. So imagine if you and I could stand next to each other and somehow swap hair color with each other. It's kind of like what these are doing. That's probably a little bit overly dramatized, but it's basically swapping genetic traits in real time and not passing them on to your offspring like you traditionally think about. It's not weird to say if you have red hair, you would pass that on to your son or daughter, and that they would have red hair. That's something you'd totally expect. That's vertical transmission. Horizontal, let me put this up here, horizontal is not what you typically think of, where two people could somehow hold hands and swap hair color that way. So if you and I stand next to each other and you have red hair, you could swap your red hair with me. Now obviously we can't do that as humans, but as bacteria, they're basically doing something along that line. Okay, they don't have hair obviously, but they're transferring traits back and forth in real time. So that's called horizontal gene transfer. It doesn't happen in most organisms, and it's pretty unique in that sense. What it does is it allows for new genetic information to be transferred. Okay, so that's another way that bacteria get resistant to antibiotics. They actually swap they swap antibiotic resistant genes with each other. So when one bacteria becomes resistant through natural mutation, it can even then go on to spread that information to other bacteria, and they call it amplifying the resistance. So there's 3 ways that this works. One's called conjugation, one's called transformation, the other's called transduction. And I've got 3 figures here, so I'll go through these kind of quick, and then I'll put the last review questions on here, and then we'll be finally done. Okay, so conjugation is where we have 2 bacteria that link up and swap their plasmids with each other. And that's really what I was showing here in this diagram. So to clarify, I was really showing horizontal transmission in the form of conjugation. Now, one thing I didn't show is that this usually takes place with what's called a conjugation pili. And we talked about these all the way back in chapter 4. But a conjugation pili is basically this bridge between 2 bacteria that allows them to swap this DNA. And this DNA, by the way, is actually referred to as a plasmid. So that's the way that works. So that process is called conjugation. One of the more common ways that it works. So they link up and they swap the DNA back and forth like that. The next one is called transformation. Transformation is a process where when some bacteria die, their DNA actually gets fragmented, and then living bacteria can actually pick up some of that DNA and incorporate it into their own DNA. And they do this as a kind of like a scavenging. It's like almost going around and picking up your neighbor's junk and keeping it. And the idea is that occasionally they pick up extra good stuff, and that actually enhances their survival. And theoretically, they could also pick up bad DNA, and it could kill the cell here. But the idea is that there's so many bacteria in the population, there's trillions of them, that if some of them die as a consequence of this, then it doesn't really have any effect on the population. But if some of them get a benefit from it, and then those go on to replicate and survive better, then that can enhance the population. So while the chance that it can kill you is definitely real, the chance that it can benefit the entire group long term outweighs that risk. And so this is a process by which bacteria are kind of just randomly picking up DNA and trying it out and throwing it in their cell and see what happens. And over time, this can lead to positive mutations and positive changes in their DNA. So that's called transformation. It involves the uptake of what's called naked DNA that's come from a cell that's died and is just floating around and gets picked up. The last part is called transduction, and transduction involves bacteriophages, that type of virus that infects bacteria. Recall from chapter 6. One of the things I said in chapter 6 is that bacteriophages can actually make bacteria more pathogenic by causing different changes. I didn't really get into a lot of detail, and I'm not going to get into that detail here, but basically what happens is a bacteriophage, inserts its DNA into the cell, and then later that DNA can be picked up by another phage along with some of the bacterial DNA. So as these and then transferred to the next bacteria. So as the bacteriophage enters, it can basically mix up its own DNA with some of the bacterial DNA, and then go on to pass that to another bacteria, and then pass not only the phage DNA but the bacterial DNA as well. And remember that sometimes these phages go dormant and the DNA is incorporated into the bacteria. So occasionally what happens is a phage comes along, it picks up DNA from one bacteria and then transfers it to another bacteria where it goes dormant and causes a permanent incorporation of the first bacteria into the second. Again, not likely to happen, but when you have trillions of these bacteria and trillions of these phages, these bacteriophages, it happens based on sheer probability alone. It's a rare event, but with all the numbers, it happens semi-frequently. So phages picking up DNA and transferring them is what's called transduction. Okay? That ends our very long discussion here of genetics. So here's the last set of questions for you. As always, feel free to get a hold of me. Let me know if you have any questions. And I will see you next time we meet. Have a great day.